This is my roadmap project, the model of the pig. The pig is an autoenzymatic digester, and their main food is corn, soybean meal, which contains sugar, starches, and fibers. This is done by their mouths, where prehension and mastication decreases the particle size of their food and increases surface area for enzymes to work. Salivation takes place where salivary amylase breaks alpha-1,4 linkages, starches, and this only happens in pigs, humans, some avian species, and it is not present in horses or ruminants. And then the food is gulped by deglutition and through the esophagus, which leads to the stomach, where there is HCl acid by chemical H bonds. There is no enzyme since it's an acidic environment, which denatures them. And then we go into the small intestine, where the duodenum is the primary site for starch and sugar digestion. It buffers amylase, and the pancreas secretes pancreatic juice, which buffers secretin and enzymes. Secrete into duodenum from pancreas buffers increased pH of chyme, exiting stomach. And this is the brush border, where there is amylase, sucrase, lactase, maltase, isomaltase. These are all single sugar glucoses. And then these are the main organs in the pig, the jejunum and the ileum, where absorption takes place, the active transport and passive diffusion. Then we go into the large intestine, where there is microbial digestion done by bacteria, protozoa, and fungi. This is known by the leftovers, where there is starches, sugars, fibers that are digested microbially, and volatile fatty acids are byproducts of the fermentation. And then the microbes use this for their own growth, produce gas and VFAs, which are the main energy source of animals, 50 to 80% of their daily energy requirement. And the three major VFAs are acetate, propionate, and butyrate. Then we go glucose, where this is where muscle cell growth. So one glucose, this is where glycolysis is done in the cytoplasm with no oxygen. And this is where glucose is turned into pyruvate. And it's anaerobic in the cell cytoplasm. And the main processes of glycolysis are activation, where there is -2 ATP, cleavage, 0 products, and oxidation, 4 ATP and 2 NADHs. Then pyruvate goes if no oxygen, lactate, and then with oxygen to the mitochondria, which acetyl CoA plus 2 NADHs times 2 minus CO2. And then this goes into the Krebs cycle. The Krebs cycle turns twice. Inside it's 3 NADHs times 2, 1 FADH times 2, 1 GTP times 2, and 1 negative CO2. These are savings of the TCA, also known as a Krebs cycle. This is where substrate level phosphorylation happens, -2 ATP plus 4 ATP plus 2 NADHs. And then the electron transport chain and oxidative phosphorylation leads to ATP synthase, where NADH, FADH leads to ETC/OP, negative negative energy, goes to ATP synthesis, where ATP is oxidative phosphorylation, which gives the pig its energy. And this is the end of my roadmap.